All right, we're back. Take it away, Ilya.
All right. Thank you, Michael. We're back for part 3. And to finish up some of our data points here, there's another problem with the idea of the modern synthesis. And this one is actually very fundamental to the theory and the thesis that I'm trying to put forward as a replacement, and that's the prevalence of promiscuous gene swapping between organisms. It's called horizontal gene transfer. So this is a phenomenon that occurs is mainly thought to have been a phenomenon common to bacteria, prokaryotes. But we shall see that there's ample evidence for its existence in eukaryotic and even complex, larger multicellular organisms. So it occurs through various mechanisms. At least among bacteria and archaea, it is extremely widespread. So in many, many branches and species of these kingdoms, we see clear evidence of genes going from one unrelated species to another. It occurs by various mechanisms, including conjugation, which is sort of direct bacterial sex. There is transduction through phages, so viruses that infect different bacteria and different bacteria species also have the indirect benefits toward the organism of spreading particular genes. And something called competence, which is quite an amazing ability. It's actually just the ability of bacteria to absorb free-floating sequences of DNA. And it's specific. I mean, it's not specific, but there is a specificity to it, meaning that they're able to tell particular sequences or tracts or pieces that the bacteria will recognize as, information rich, or have some sort of receptor, basically be able to tell kind of what it is before it takes it in. So it's not a non-specific thing. You don't want to, it would make sense. You don't want to absorb any kind of random random genetic sequence. You want something that is interpretable. And of course, as we've noticed, stress and DNA damage are actually stimuli towards increase in this competence. It enhances this phenomenon in bacteria. So what does this mean? It shows that there's definitely an ability for bacteria to be able to patch their DNA damage or be more interested in adapting to stressful circumstances through direct modification of their genome by the most direct mechanism possible, which is literally absorbing it from the environment.
Which again indicates an awareness of what the meaning of the genes or the DNA that they're absorbing is in order for them to make any use of it.
Yep, absolutely. That's the point we keep coming back to, and interesting fact. Let's see. So the question, of course, arises, as I mentioned, that there is evidence for eukaryotic organisms being able to transfer genes and take in DNA from other species. And particularly interesting is the evidence in human beings. So this is sort of an aside because these aren't the most glaring or colorful examples that I could find, but it's kind of interesting to think about. So although there are many interesting genes that are historically within the vertebrate or mammalian and primate lineages thought to be the product of horizontal gene transfer. So specifically, the mechanisms of the vertebrate adaptive immune system, according to one researcher, Michael Deem, D-E-E-M, there actually are human somatic cells, so our cells that are in our body, not necessarily our germ cells. So this wouldn't really affect evolution per se. But it seems that our somatic cells are actually able to trade and share DNA. So when one cell goes apoptotic, where it has programmed cell death, it falls apart into these specialized blebs and for easy kind of absorption. And it shows that neighboring cells actually incorporate the debris that carries the DNA from that apoptotic cell and are able to basically take it and use it. And so if you label these cells, when they apoptose, neighboring cells will take up and incorporate that DNA directly into their genome. So
that's interesting that the cell, it sounds like the one cell will dissolve in a way that actually does make it easier for them to be absorbed by the neighboring cells. So that
yes.
Yeah.
In fact, there is evidence. It is medically relevant because now most blood that's given is what's called leukoreduced, meaning they get rid of the white blood cells that are transfused. So you don't want someone else's white blood cells in you for a lot of reasons. But if you give whole donor blood, which is kind of done in extreme emergency situations still in parts of the world where leukoreduction is more difficult, and was used to be done much more often, it seems that even after 10 years, there is detectable in the blood of patients who've received someone else's white blood cells. Their own white blood cells will contain about 2 to 5% of a patient's white blood cells who received someone else's white blood cells in this kind of whole blood. They will have some chimerism, so they will have some trace in their own white blood cells of this foreign DNA.
They cannibalized the DNA to put it in a dark way.
Yeah, that's very kind of disconcerting. I guess especially if it makes you think of those Jehovah's Witnesses are onto something.
Oh, I see.
But no, it is interesting to think about. And there's even evidence that cancer cells actually incorporate portions of bacterial genomes, as crazy as that sounds. And even at the level of the mitochondria. So there's some evidence that certain kinds of bacterial RNA transcripts have been found in human cancer mitochondria. It's kind of weird. Not clear if this may be just a series of errors, although the data on which it's based seems reliable, but there's really no understanding of what significance this has. Although it's known that bacteria are carcinogenic in other species. So like in plants, there are specific bacteria which form tumors, interestingly. Now, and actually, I mean, we do see that. Let me correct that. We actually do see that in mammals. Bartonella will cause is a carcinogenic bacterial infection. So is H. pylori. It can actually cause a kind of lymphoproliferative soft tumor in the GI tract. So that's not entirely true. Point that out. Unclear if there's a genetic mechanism for that, but that would be interesting question. But it's even hypothesized that, as I mentioned before, mammalian placenta uses repurposed viral genes. And so that's the question is how did that happen if not through direct germline infection of a particular virus? there's also evidence that older siblings, so in sequence, will pass on their at least a tiny portion of their DNA to younger siblings via fetal stem cells that are left in the mother.
Oh.
So a woman will actually get circulating fetal stem cells every time she's pregnant. They have some interesting medical effects. It's been known to modulate autoimmune diseases in pregnancy, and may be an explanation just for some of the curious phenomena that we see regarding autoimmune diseases in pregnancy. Sometimes it gets better, sometimes it gets worse. There's even cases of cancer being kind of suppressed during pregnancy. Some of this is theorized to be due to these circulating fetal stem cells that women get. There is also, in at least in fruit flies, this is definitely the case, and it may be the case in other animals, that when internal fertilization happens, so it's possible that that there is some kind of ability to pass on traits from previous male partners to future offspring. So if an animal, in this case the fruit fly, had eggs from one male, and then subsequently, next season, or I don't know how long mating, probably much shorter than that. The next male that mates, some of those offspring actually have traits from the first male, and they actually have DNA from the first male. It's unclear how that happens exactly, because fruit flies don't really store sperm in the way that certain animals, certain insects especially, do. But there is something maybe to this, ancient Aristotelian theory of phylogeny, and that there may be an effect of previous reproductive cycles on future ones. Anyway, it's weird stuff. But what's much more obvious are actually some of the horizontal gene transfer effects seen in other eukaryotes. So here are a few organisms out of many, 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 many examples. Many examples that I could pull up that have known horizontal gene transfer in eukaryotic cells. These are, the complex cells that are characteristic of multicellular organisms. So one is the coffee borer, and this research has been done because it's a major pest of coffee. And it has a gene called HHMAN1, which encodes a mannanase, an enzyme that breaks down mannose, which is a mannase, rather, a type of sugar found in coffee. So the protein, according to these Brazilian researchers, is clearly just taken and put into the genome, and it's from bacterial origin. So this insect is able to produce an enzyme that's clearly from a bacterium, which is interesting. How did it possibly acquire this? It's very weird. Now, there are many fungi, especially those that form a symbiotic relationship with other organisms, like in lichen, for example, that are actually loaded with bacterial proteins that come from bacterial DNA sequences that they have. Specifically these polyketide defensive chemicals, these sort of toxins that they produce to avoid being predated upon. There are the Rafflesia plant, these giant stinky flowers that are the biggest flowers in the world. They're famous for being smelly and kind of interesting. pollinated by flies.
I
It's
am
actually,
familiar.
yeah, yeah, they're a parasitic plant. So they don't have leaves or stems, or they just bloom. It's kind of almost like mushroom-like flowers, but they're actually living inside these other tropical vines. Up to 2% of its transcribed genes are directly stolen from its host. So that's rather remarkable. And this is actually true of many parasites, actually. And these genes don't just, they're not just non-functional. It's not like this is some sort of virus or maybe something that threw it in accidentally. Most of them are functional and have actually replaced the functional proteins that this plant originally seems to have produced. So it relies completely on the stolen DNA to function.
Interesting.
Even though it's a small portion. There's what I think is one of the craziest examples is this sea slug that's portrayed here, Elysia chloritica. What it does is it eats algae, and it actually absorbs the chloroplasts from the algae that it eats and stores them in its tissue, so that it actually becomes photosynthetic itself. Okay?
I remember you mentioning that. Yeah.
But what's interesting about it is
Oh
that
no, you didn't mention that. I heard that on some nature program or something. Now that I
that's
think
not
about
all.
it.
So I mean, that's kind of amazing in itself. But what's
Yeah.
more interesting is that chloroplasts are like mitochondria. They're thought to be these endosymbiotic organelles that they have their own internal DNA. They're probably an ancient kind of devolved bacterium. It has its own DNA, but chloroplasts have to, in order to function, they rely on sequences, DNA and proteins that are actually found in the nucleus of plant cells. So just like our mitochondria wouldn't function without some of the nuclear DNA that we have that supports the mitochondrion, chloroplasts can't function without these nuclear part of its information stored on the nucleus, like it is in a plant cell. But here's the truly amazing part, is that this animal has some of these required genes in its nucleus to keep those chloroplasts functioning. It's the only animal that is known to have them.
Mm-hmm.
It couldn't, there's no related sea slug with, protogenes or something. There's just no, there's no animal that has these. These are 100% plant algal nuclear DNA.
Mm-hmm.
It clearly must have gotten them from its food as well as the chloroplasts. And that's the most likely explanation. I mean, we could think of other possible explanations, but this is likely an example of horizontal gene transfer from a plant to an animal, of all things, across pretty wide evolutionary gap.
Yeah.
Yeah.
So again, all of these examples, they mess with the traditional model, which is supposed to just be from the bottom going up. But if you've got horizontal transfers, all of it is just again and again examples of there being this taking control of the DNA of the organisms. There being, like I keep saying, the double causation. And the animals sort of messing with the ability of the traditional notion of evolution to sort of push everything forward in front of it. Animals are smart and are figuring out how to hack their own DNA and stuff in all these crazy ways.
Yeah. I mean, there's numerous plants that have done this. As I mentioned, there's an arborella vine. There's Wolbachia, which is one of the craziest examples of bacterial parasitism / symbiosis in invertebrates and insects. I mean, that really deserves a whole, it's probably actually is the subject of specials on various biology programs, but this is a common endosymbiote / parasite of many insects that really completely messes up the life cycle, reproduction, and really sex determination. basically can turn all the offspring of an insect into females to ensure that it gets passed down. And there's ample evidence of gene exchange from this bacterium to its arthropod hosts. And there's examples of arthropods and insects. Basically, anywhere that you have very tight, the most obvious, easiest to find examples of horizontal gene transfer is where you have very tight ecological interactions between different species. So anytime you have that, you're almost guaranteed to find a degree of horizontal gene transfer. But really the one example that actually deserves its own slide here, because it's probably the one that demonstrates the biggest gaps. So we saw the plants, we saw some bacteria to insects, we saw some plants to animal, one example, with the Lesia chloritica sea slug. But these lovely ladies are Bdelloid Rotifers. And they are probably the most prolific and promiscuous horizontal gene transfers. I guess they're acceptors of DNA that's known so far in biology, actually. So it's a group, these Bdelloid Rotifers, that includes about a few hundred species, and until recently, it was actually thought to be asexual, and actually this was thought to be the largest group of asexual organisms. As I mentioned before, asexual organisms are kind of rare, kind of recent evolutionarily, and for that reason are thought to be basically broken sexual organisms that are kind of an evolutionary dead end. What's interesting is that this was considered to be a very large, well-differentiated, well-speciated class, and it was asexual, largely. And this was thought to be strange. Well, it turns out that when they actually studied the genome of these Bdelloidii,
just to be clear, what exactly? I'm not quite sure what I'm looking at. Oh, I keep putting my finger to the screen. What are they? Are they like worms or little?
They're rotifers. It's basically kind of plankton. They're
okay.
They're a tiny, they're a tiny kind of arthropod-like, primitive zooplankton. They have little mouths with little filaments and parts. Skeletons. And
just wanted to clarify that. I was like, are they some strange creature's teeth, or I don't know what I'm looking at. Plankton, that's good.
Yeah, there's a kind of zooplankton, basically.
Yeah.
I think they're found in both fresh and salt water. But so they sequence their genome. Now, what's interesting about their genome is it actually contains basically an archaic set of non-functional chromosomes. So it actually has 4 different chromosomes. There's 2 different sets of chromosomes. There's a pair that are basically thought to be archaic and non-functional. Already that raises red flags for me, right? I mean, why on earth would an organism keep this set of chromosomes if they were completely non-functional?
Right.
That's what they said. And so they have one diploid set. Remember, diploid is like, how many pairs it has. So it has one diploid set that's non-functional and one set of chromosomes that's functional, technically making this organism tetraploid, meaning it has 4 sets of chromosomes. But allegedly it's just functionally diploid. So it was found that when they studied them, that they actually somehow ended up undergoing recombination and exchange of alleles, just like animals that have sex. The scientists hypothesized that, kind of like some related types of rotifers, they rarely, under special conditions, would spawn males, and then that would allow genetic recombination. Although this has never been, these are like pretty well studied, has never been observed under any conditions that we know of, maybe somewhere in the wild. But furthermore, what they found, which was really the most bizarre part, and the reason I put them here, is that the genome of this animal, the functional half, that is, included 8% non-animal genes. So like straight up sequences that look like they're from plants, fungi, or bacteria, especially in the areas like at the edge of the chromosomes next to the telomeres, right under the telomeres. They actually contain the largest diversity, but fewest number, if that makes sense, of transposable elements of any known genome. So remember, we talked about these transposable elements being like the basic building block of genome complexity. This thing had more different types than any known previous genome ever sequenced, but they weren't like humans. We have lots of repeats of the same kind. This had the fewest actual
repeats.
Yeah. And so they were baffled by this finding, which was really bizarre. And one theory, so they came out with this theory that what these animals do is that they actually, so they live in an environment where they often dry up and then get wet again, dry up and get wet again. So they're like found in like puddles, basically.
Right.
So they go through numerous like desiccation and rehydration cycles. Now, what ends up happening is that when they undergo these cycles, their DNA gets damaged. Hold on, excuse me for a second.
You're okay. Here, I'll pause the recording. Okay, we're back.
All right. So these foreign, it's thought that all this extreme presence of these foreign genes actually represents the organism absorbing just random DNA from the environment to patch its genome after these desiccation, rehydration.
Oh, I see.
And it's not just there to, to look pretty on the ends of the chromosomes. It actually turns out that some of these foreign genes were transcribed and they produced functional proteins, even associated with some important like basic regulatory sequences that involve like the body plan of the organism, detoxification enzymes, antioxidant defense, genome repair and regulation, and even digestive enzymes. So some of its basic cellular function is all borrowed from whatever DNA it can pick up from the environment. And what's interesting is that these rotifers are over 10 times more resistant to radiation than an oxidative stress than related species of same rotifer, similar rotifers. So this is like crazy. It's like an X-Men, like mutant power. What's that
right.
one? And like touch other mutants.
Rogue?
Yes. So these things are
they're
able to absorb,
other species powers or something.
It's that bizarre. So yeah, again, 8% of its genes are not even animal. And actually, if we look at its actual functional like enzymes and sort of the genes and proteins that deal with interaction with the environment, meaning response to stress, response to toxins, ability to deal with food, 40% of those seem to come from this mechanism of horizontal gene transfer. It's quite amazing. I mean, what else is out there? Right? I mean, this is stuff that's literally underfoot. Let's see. Let's go on to the next one. What's this indicate? A little. I don't know. We'll skip that. No, it's a faux pas to not have images on your PowerPoint that you can talk about, but that's okay. We're already running long. So let's look at kind of what this all means. So zooming out, right, kind of going back to our original thesis, we're talking about a broader perspective on life. And a lot of this research seems to come from people who are not traditional biologists, but physicists, statisticians, bioinformatics, people in molecular biology or molecular genetics, biochemistry. The work of the late, now Eshel Ben Yaakov, who was a physicist, Israeli physicist, who took an interest in the behavior of bacteria, sort of in the wild. He, instead of studying bacteria in ideal conditions on ideal growth mediums or ideal solutions of food, he sort of got the idea of how do these biological systems behave in situations that are more natural, that have obstacles, that have stress. He was interested in sort of the behavior of these systems. And what he discovered actually is that bacteria display some really amazing problem-solving skills and abilities when they're exposed to environmental stressors. And in the wild, they often live in colonies, right? Bacterial colonies, biofilms that are really in complex interaction with other species. And they often have very promiscuous exchange of DNA, usually via conjugation, transduction, even competence, as we mentioned before, these mechanisms. The behavior requires a degree of communication, integration, and understanding, right? So if you look at the individual bacteria, it may not kind of know its entire role in the colony. But if you look at the colony itself, it's able to perform a lot of very complex tasks, the way it grows. And
kind of like the human body.
exactly. So if you don't put something into the stress that it's biologically necessary, it won't really develop its full potential. And what he found was that different bacteria species and different colonies have sort of different intelligences, so to speak. They're different. Some are very exhibit sort of complex problem-solving behavior, others really don't as much. And he began to think of the colony as a kind of single evolving superorganism structure, because oftentimes these organisms are clones of each other, or at least very closely related as they progress through time. And here in this orange-yellow picture, we see demonstrated a phenomenon that he basically discovered. It was never previously, I don't think, described, something called sibling lethal factor. It's basically two identical colonies of bacteria that was producing a toxin or a repellent that's purely aimed at other identical sister colonies, so that they don't grow too close to each other. And this is just one example of the bacterial communication and signaling that he began to study. The kind of signals that bacteria produce aren't just for other members of its colony or other colonies. They're for other species, and even other, can even interact with other kingdoms. So for example, besides the idea of sort of toxins, everyone can kind of get that idea. You're trying to kill off. It's sort of a very primitive way of communicating, I suppose. But there's a way that bacteria can actually interpret the signals of eukaryotic cells. For example, there's something called, that's well known in medicine, it's called norepinephrine enhanced virulence in many pathologic bacteria. So like a species of Staphylococcus will become basically more aggressive when it senses that there's adrenaline, epinephrine in the tissues, meaning that if an organism is under stress, it knows that the immune system's down and it's time to go ahead and kick off. The same thing is actually seen in herpes viruses. They have a sensor, so they become latent. Many herpes viruses, like the ones that cause shingles, like right late in life, they kind of that cause, cold sores. They're latent in the nerve cell or wherever they go, usually some ectodermal cell. And then they will sit there and wait for their sequence that's lodged in the genome actually has a sensor for cortisol, or the common stress hormone cortisol. When that is elevated, it actually binds it and gives a signal to that virus that it's time to, again, get started and replicate because the immune system is down. I mean, people who have, everyone knows this. I mean, you get cold sores or even a shingles outbreak under periods of stress.
Mm-hmm.
So there's a very direct sort of explanatory mechanism there. There's a degree of communication where there's sort of organisms under stressful conditions can sort of decide to sporulate or form either reproductive spores, which are more resistant to environmental conditions. And basically the last one to do it benefits from everyone else kind of popping off and leaving more resources. So there's kind of a game theoretical communication
Right.
inside these bacterial colonies. And Ben Jacob found that they will actually undergo sort of stasis. These bacteria that are under sufficient stress will pause their growth and reproduction, but they will still sort of maintain their genome in an activity which causes mutations. And that actually can sometimes go up in certain individual bacteria that are kind of like hypermutable phenotypes. And that those will actually result in a breakout from the stressful static situation that the colony finds itself. So they will take it upon themselves to kind of heroically kind of sacrifice possibly fitness to a prior environment in order to overcome the barriers of this new dangerous environment. And they will be the ones that break through sort of to the other side of this technical hurdle. This kind of behavior, he posited, requires something called genome learning. He used that quote. You need a spatially and temporally variable environment, the ability to remember the past conditions of the environment, right? You have to have a self-awareness of the genome structure and its capacity for change. You have to know what to do. And you have to have the means for the organism to recognize these problems and to formulate a sort of a model or a solution to this problem based on the collective information and the processes and its internal and external conditions. So it really has to have a degree of awareness of itself and the environment to a great extent. And finally, it has to have this, what he called cybernetic capacity. So the capacity to actually change itself based on these solutions, including gene modification. He asserted that this kind of bootstrapping by sort of, instead of, this isn't like, a bacteria all by itself. It works through the information that's collected and filtered down through the whole colony. So there's an emergence that occurs from the collective intelligence of these bacteria, and it emanates down into the individual bacterium, and it's able to act in a way that's greater than just the sum of its individual parts.
It's like the internet.
Yeah, it's like a lot of things.
Oh, sure. Yeah.
It's exactly that. It's this kind of behavior that comes around because of the interaction of its parts.
Yeah.
But in this case, it exceeds the computational capacity of the individual bacterium.
Right.
It's completely non-trivial. And in fact, A similar model was proposed by one Tordé, yeah, Torday. He is a researcher an evolutionary biologist who theorized on the mechanisms of what are called internal selection. He was of a group that pioneered the discovery and development of corticosteroids for lung maturation in premature infants. His question was why? Why on earth would corticosteroids have anything to do with lung maturation? And he basically came out with this theory that it all goes back to the development of life, the capacity of life to live on land. And that corticosteroids not only develop lung tissue in a way that is conducive to life on land, but they also cause bone and cardiac remodeling, and epinephrine, other kinds of stress hormones, not just cortisol, not just corticosteroids, but the adrenal hormones, the other adrenal hormones, they also are involved in the kind of cardiopulmonary remodeling that's conducive to life on land. And his basic hypothesis was that the struggle, literally the physiological struggle of an organism attempting to breathe and live on land was enough to induce certain changes in its internal physiology in a way that's similar to how stressed bacteria kind of break out and find new solutions. The actual internal environment of the individual cells provided that milieu to have like a severe but non-lethal stressor that eventually got passed on. So he is explicitly Lamarckian, but what he says is that this is a form of natural selection that occurs not at the level of the whole organism, but at the level of the internal environment.
Right.
So that's an interesting way to think about things.
Yeah.
This is kind of a summary. I don't know, it's kind of blurry on my screen, unfortunately. But this is a chart that I kind of made going back to our original ideas on different evolutionary theories to finally summarize some of the things that we discussed. Or actually, we haven't talked about Baldwin and Waddington, but that's for completeness sake, they're included. We're dealing with two issues, right? How do we get from a changed environment to evolution, to sort of adaptive, inherited change? And there's several ways we can go. We can talk about the effect of existing variation and its interaction with natural selection. That's sort of the conventional Darwinian thought, although, as I mentioned before, Darwin wasn't opposed to other mechanisms, in fact, proposed other mechanisms besides existing variation. We have what's called Kimura's neutral mutation theory, the idea that things sort of drift along without any particular pressure, and that's actually the main driver of speciation. We have several other theories, including the direct response to stress resulting in internal selection, and then followed by sort of genetic encoding. This was Torday's theory. And then we have stress resulting in sort of direct natural genetic engineering by some of the mechanisms potential mechanisms that we outlined earlier. And I think I do talk about Baldwin, but that the idea of learned behavior is the idea that the environment changes, the organism learns a new skill, like at a very high abstract level. It's behavior.
Yeah.
And then
like using tools or something like that.
Tools like, yeah, feeding in a particular way, a different pattern of collecting or whatever, washing its food, I don't know, hygienic behavior in bees, and then that then constrains, well, bees maybe learn more genetically, but that's irrelevant, neither here nor there. And then that learned behavior will then go down and constrain the type of selective pressures, of course, that occur. So there's definitely a feedback loop between learned behavior and the trajectory of evolutionary development. And of course, there's classical, what we might call Lamarckism or neo-Lamarckism. The needs of the organism change, its behavior changes, and then there's a use or disuse of structure that then is passed down somehow. So that's the points I want to talk about here. Now, when we consider a reductionist materialist approach in biology, and we don't think about a biological system as anything more than a computational system, we're actually doing it a great disservice, because if we consider the actual computational models that are in existence, and by this I mean what Turing called the universal Turing machine, right? This is it sounds like a pretty abstract and maybe even primitive model, but it basically is a computational thought experiment that describes technically all computers, all non-quantum, I should say, all non-quantum computers, and any program that can run on it. And this rule consists of nothing more than reading and manipulating symbols on an infinite tape. Now, the symbols don't have to be ones and zeros, it doesn't matter. But a universal Turing machine is really sort of a construct that has, again, there's an infinite tape, and it reads and modifies the symbols on each square of this infinite tape according to a program. And that program is
algorithm.
A set algorithm, a program, yeah, whatever you want to call it. And it's nothing more than the instructions on moving and modifying the symbols on each square of the infinite tape. All computational devices, as I mentioned, except quantum computers, map onto this basic model. And what's amazing is that you see in the literature, in the biological literature, people seriously saying that, hey, we should think about organisms this way. And if we think about organisms as, quote, nothing more than biological robots, as, what's his name, Daniel Dennett likes to think about, we will have to shoehorn a lot of phenomena that don't fall and literally cannot be described by such a Turing machine, as we won't be able to explain them, and we'll be throwing them out of biology entirely. So this common thought cliche that maybe is less common now, but certainly existed in the 80s and 90s, throughout the 70s in biology, and really strangles the field to this day, is fundamentally wrong, because all of life's recursive complexity and the ability to change the underlying rules and structure, as some of Ben Jacob's work kind of outlines in his specifically his papers, where he demonstrates why these bacteria are nothing like Turing machines. It really cripples our ability to think about this. And to give you an example of some of these quotes, so metaphorically speaking, the genome includes a user with a computational unit and a hardware engineer with a team of technicians for continuous design and implementation of changes in the hardware. So such a complex is far beyond a universal Turing machine. In the latter, that is to say, a universal Turing machine, the structure is static and is decoupled from the input and output and the computational process. This is very important. The structure is static and is decoupled from the input and output of the computational process. Okay? So the Turing machine can't decide that it wants to run a different program.
Right.
Exactly. It can have a meta program, which then does this. And of course, that's kind of the thing that biologists who are still clinging to this model will say, well, you know, this is just natural selection, and this is just the mechanistic effects happening on a different level. And you can keep doing that, right, up to a point. And then eventually what happens is that you get into the issue that science and empirical studies, or even non-empirical mathematical systems, are underdetermined. That is, you can keep appealing to a higher level meta framework, but that just goes ad infinitum.
Yeah.
And that's what Gödel proved, even in mathematical systems, where any non-trivial system of axiomatic system, like a really, you know, a mathematical system where, you know, 1 = 1, it ends up being in a situation where you cannot make it both complete, you cannot prove everything within its framework, and yet at the same time consistent, meaning that, you know, you can get 1 = 1 every time. So the inability to have both of those situations met is a real problem for the materials reductionist perspective, that things have to be sort of nice, tied up in a bow, causal and knowable. It's the sort of the conceit of sort of this kind of Luciferian worldview, where rationalism will be able to get us out of this, you know, this fear of the unknown. Yeah, I mean, if mathematics is plagued by this, then I mean, there's no firm foundation to stand on for science, which can be shown to be underdetermined in much simpler ways, actually, relying on Gödel's theorem. I mentioned before, I think, in the first part of this talk, the Duhem-Quine thesis about underdetermined nature of any theory. Now, these logical paradoxes that life seems to weasel out of isn't really because it's, you know, it's just sort of our illusion, because we're relying on models which are dumb, which don't apply to biological systems. No, I mean, these models are useful in computation, in, you know, technical fields, but they are not the only way to look at the world. And they're not a way to model systems which don't behave this way. And I think I've at least pointed out, like, I can't prove, it's something that's hard to prove in the sense of scientific proof, right? You can't, even natural selection, you could argue, is difficult to prove in an environmental setting. Theories of evolution are mostly speculative. They're not testable theories in the scientific sense, or at least not easily.
Yeah.
And because of that, they don't really meet the technical Popperian criteria of falsifiability. Nevertheless, what the purpose of all, you know, this list of various observations, this kind of weird genetic phenomena that I rattled off, they're individually, I'm not proposing some direct mechanism where I can explain anything. No, the purpose of this is just to show that it's much more easily interpretable and understandable, all these things together, if we consider life to be self-evolving, essentially. Life to be self-directed, self-modifying, a self-genetic engineering thing. So this view is much more useful, right? It's
You have to reintroduce will into the system, in a sense.
Yep, we have to have a different model of the system, one that has a telos, a will. Yeah. It's not always clear what this will is, because it's hard for us to put ourselves in the subjective environment of a particular organism. And here you have, the Terminator.
Yes, yes.
But what I mean to show is that an artificial intelligence cannot occur based on the principles that that a Turing machine works on.
Yes.
It would just not be at all like life, and not because specifically because life has this inner will, whereas the very processes that a machine works on, even infinitely, you can make a machine as complex as you want. It's still not going to have these recursive self-awareness and an agency that an organism seems to get from the bottom up. Where this comes from.
Mhm. I was just going to say, you can get the same effect if you do a thought experiment along the lines of in order to remove intentionality or remove will from a system. If you take, say, there's actually a short story it was from like the 1980s by a Russian scientist who talks about how this guy gets like a whole stadium like of 1,000 people together, and he organizes them in such a way that they all have like a little tiny bit of instruction. Like they've been given a set instruction of what they're supposed to do, and then he just sends paper in one end, and they basically just do all these mathematical operations that basically simulates a computer. It's simulating a Turing machine, and the paper goes through. And the point being that it's like, okay, every single one of those individual humans, obviously we would describe as having will and stuff, but each one of them there's no understanding magically from sort of the higher end. You organize them in a computative way that does not magically a consciousness make. So they would make stupid mistakes that you see this in video games all the time. Like you see video game glitches that people make fun of where those are entirely a result of the machine has no contextual understanding. It has no actual goals. Everything is just rule-based. And so you'll get, if everything works out, then it can look kind of like real life if it's complex and contrived enough. But if you get a single glitch, then things will walk through tables, or they'll like keep shouting at you even though they've just been shot and they can't do anything. Like all these sorts of things that make no sense in real life, because it can sense the context, and they can sense the context because they have a goal that they're moving towards. So everything falls into place around them. And that's what a machine or a Turing machine can't do, for the same reason that a stadium of people, each one having this individual little instruction that they have no idea how it fits in. It's basically the Chinese room of John Searle. Yeah.
Yeah, that makes sense. Yeah, it's kind of like a proof by negation of this idea. Yeah, it's a lot like that. And to have a model of the environment, to have context, one has to have an agent that has an internal sense of awareness.
Yes, that has a point of view.
Right, a context is only meaningful. Yeah, this is where we get to. So my next slide actually talks about this. So this context is only relevant for an agent that is capable of having context, right? So this is the difference between natural and artificial, one could say, thinking or computation. For an artificial system, there's input, output, and the control. There's a direct, if you view an organism this way, it's just a simple exchange of energy and waste with the environment. The genetic code is nothing more than a program. And the output is sort of a self-assembly, duplication, and various search algorithms to complete things. And there's a degree of there's this overarching what's called finite control, and that the only source of variation are these reproductive errors. So the way the system is not working completely 100% is the source of this evolution. And then there's this ruthless selection based on just the environment. This is really the conception of evolution in the sort of public consciousness, even of fairly educated people, which is quite remarkable. So, champions of materialistic philosophy, Crick, Dawkins, Minsky, Daniel Dennett, most of the really famous so-called skeptics claim exactly this, that humans, are nothing but, quote, biological robots, or some other metaphor that reduces the complexity of human beings to these material interactions. But we've, as I hope I've demonstrated, even the simplest organism exceeds the basic functional and computational limitations of any computer, which has, a program script, as I mentioned. No concept of self-identity, no ability to write its own programs or modify itself. So a change in the environment has no meaning or relevance to a Turing machine in the way that it would for a living system. Now, if we think about living systems, their actions are goal-oriented, teleological is the old term. And they're geared towards, you could ultimately say survival, but that's not quite the right term. There may be more to it than merely survival.
As Nietzsche would say.
Yeah, yeah, and this is why we would say throwing, our instinct that throwing a puppy into a river and throwing a cell phone into the river are two very different acts.
Yes.
So equating them is, I mean, I wouldn't even call it psychopathic. It's something different. It's just doesn't make, it's very, very illogical. It's really naively obvious, right, in our experience. Kind of zooming out and kind of talking about some of these, very basic, intuitive inferences. It's really naively obvious that a living system cannot be dead like a machine is. Simply, really, the existence of our own subjective experience of reality demonstrates that we are different from a universal Turing machine. I think you go through this in your thesis that you read. I think it's very interesting, actually, to think about. It's be quite relevant at this point. I would, should plug your own video here.
Which video was that again? I'm trying to remember.
I think it's where you read your thesis. It's right
yeah.
Your computer.
Yeah.
And what the differences are between us and it, and kind of the sort of the train that you go through all these various philosophies and how to think about it. It's actually useful exercise, I think.
Thank you. I'll put a link to that in the description below. I wrote an essay, if anybody doesn't know, that I read on my channel. So it was talking about consciousness and some of the things we've talked about today, actually.
It's amazing enough that really so many intelligent people, philosophers, scientists, really hold on to this bizarre view, and empirically false, I would say, believe, at least based on some of the stuff that I've hoped, I presented and makes sense, that human self-awareness is nothing but an illusion of sorts. Of course, the question arises, if it's an illusion, who's being blinded and
who's, if you have an illusion, you have to have somebody for whom it is an illusion. That's why I just can't. By the way, I've read through, it was that's a part of my essay where I'm grappling with Daniel Dennett, and I've actually sat down and been like, okay, he's a smart guy, so there's got to be a way I can look at this where I don't just feel like not taking him seriously. Like, I want to be fair to him, but I have such a hard time. The only way I can take him seriously is if I say he just doesn't realize that he's actually a panpsychist. He just doesn't realize, like, he keeps saying, but his theory makes a lot more sense if he's a panpsychist, and then he just has a combination problem. But anyway, that's a whole side note thing.
No, that's an interesting point. Yeah, I mean, it is weird. You expect for someone with the head to beard hair ratio of him to be more
Now,
aware.
now. I will give him this. His beard is magnificent. Is magnificent. Good on you, Professor Dennett. Yeah,
Well,
yeah, yeah.
well, let me get on here to the rest of the slide. So talking about the biological side, you can have a comparison between these two models, and all these terms, and this comparison is actually taken from Ben Jacob's paper
Oh.
on it's called meaning-based natural intelligence versus information-based artificial intelligence. And
Yes.
here you can also see a diagram of the universal Turing machine on the bottom left. And we have a sort of a cybernetics diagram from psychologist Jordan Peterson from his maps of where he describes sort of the basic pattern of biological behavior. So it shows like a present state, right? Certain assumptions about the internal and external environment, and then a modeled ideal internal state to which an organism strives via planned and adaptive goal-seeking behavior. So the essential feature in this model is novelty. So there has to be sort of a constantly emerging chaos, right, of nature, which can only be modeled in a very limited dimension by the individual organism because it's not infinite, right? The organism is finite. So when presented with an infinite universe, right, of infinite variables and infinite categories of variables, it's only able to model these things in limited dimensions. So almost entirely, the modeling has to be limited to the organism's goals and predictions of its future states.
Yes.
And by that, the adaptation and survival is therefore an active teleological pursuit, starting at an extremely basic, as we have seen, even possibly molecularly basic. And I can't speak to quantum phenomena, but this is why I think
there's a case to be made.
Sure, exactly. But I think here it's at least more easily delineated processes in the organism. So even at the very basic molecular level at the organism, it makes sense to view these processes as teleological, and they go all the way to the highest, technical, artistic, and spiritual pursuits of mankind. Mm-hmm. There's a homology between what is above and what is below in matter. Mm-hmm. So all these attempts to everything that we do really are attempts to master, describe, model reality in nature. And the origin of novelty, oops, did I not include that? Well, we have I'll talk about sort of the archetypal symbolism here, right? You have the archetype of chaos. For those unfamiliar with Jordan Peterson's lectures, Tiamat, Echidna, the Ouroboros, other representations of combined, chthonic, maternal, reptilian symbols. And to overcome chaos, reality must be carefully observed and understood, which is why Marduk, who slew Tiamat, had eyes all around his head, why Argos Panoptes, the 100-eyed, is the one who slew the Echidna, essentially a version of the same myth, and why the falcon-headed Horus, and with his famous eye, is the one who defeated Set, the god of chaos in the desert in later Egyptian myths, and why Perseus slew the Medusa, again, a similar type of creature, by viewing her image reflected in a polished shield. Mm-hmm. So this is the idea of modeling nature in a way. All this to say that there's a symbolic correspondence between what goes on in these situations and what's going on in the mythopoetic sphere.
Yeah, in Jung, which obviously Jordan Peterson is very sort of a student of Jung, Jung talks about the notion of when a person, or you could extract it to any, or extrapolate it to any organism, when they reach a point where they can't figure out the solution to a problem because they've solved all of the easy problems, and then they finally come up against something where there are two opposite principles that they can't figure out how to navigate the situation. And Jung said that then something called the transcendent function, which is basically just the unconscious, because he had sort of a mystical view of the unconscious, but kind of like the notion of it's what would happen was a new symbol would arise from the unconscious. And by symbol, he meant something very abstract, or it's basically a model of how things are that has not fully been understood yet. So a dead symbol is something that we already kind of have completely apprehended it to the point that we don't really need the symbol anymore. It's just a part of how we operate in the universe. But a new symbol is something we haven't quite figured out yet. So he seemed to think that the mandala had something to do with that, that the mandala was a sort of symbol that the unconscious had brought up to help us to understand chaos. It was like a modeling of chaos that we're figuring out. I just thought that was interesting as you were talking.
Oh yeah, that is a good point. I also wanted to talk about here what I think is the blockage, right? So to quote Nietzsche again, so this prevailing instinct in fashion, which would much rather come to terms with absolute randomness and even the mechanistic senselessness of all events, than the theory that a power will is acted out in all that happens. So there's a resistance to this idea that right, these symbols can arise. These true novelty can arise from us. It's, I mean, you know, this is symbolized in many conspiracy theories as kind of the, or really it's symbolized best in sort of the Gnostic idea that the world is intentionally trying to delude us from our godlike nature.
Right.
And that, even so-called redemptive messianic figures were over time, suppressed and battered down with all this kind of baggage of obscurity and darkness that's meant to take away from their true messages and meanings. I mean, it's interesting to think about. To quote Ben Jacob again, I want to mention that he talks about Turing machines, and he adds the idea that the term von Neumann machine. So it's unlike an ordinary Turing machine, the program also has instructions for the machine to duplicate and disassemble itself and assemble many machines into an advanced machine. This is sort of a modification and kind of like a dominant top-level emergence view, quote, in the studies of complex systems and systems biology based on the Neo-Darwinian paradigm. So it's a patch to this
right.
old. But he, quote, explicitly proposes that the ability to assign contextual meaning to externally gathered information is an essential requirement for survival, as it gives the organism the freedom of contextual decision-making. By contextual, we mean relating to the external and internal states of the organism and the internally stored ontogenetic knowledge it has generated. We present the view that contextual interpretation of information and consequent decision-making are two fundamentals of natural intelligence that any living creature must have.
It's fascinating to me because I swear I didn't read any Ben Jacob or any I guess Ben Jacob would be the main one because he was throwing the term contextual around all over the place, but it's just like that's just a word that came to my own mind when studying typology, this completely different thing. And that's, I mean, people don't know who are watching this, that was the first thing that was like, holy crap, Ilya Dubovoy, he's on Young to Live By, and he just used the term goal-oriented, which I know it's not like a terribly unique term, but I just hadn't heard anybody use it before. And then suddenly I start finding it in the literature everywhere, and I was like, wow, it's like I paralleled some of these ideas, and it's really satisfying to see it pointed out these other areas, the notions of contextual. And anyway,
yeah. And you know, another way to think about things more broadly. So we're just kind of zooming out of the genetic, you know, microscope, and going into more like complex systems theory, cybernetics. There's this idea of recursive complexity, right? So when organisms are in network with other organisms, when they're in network with their environment, there's top-down effects that are unpredictable from the bottom up. Now, some people reject that on principle. They say that, oh, we just can't model things accurately enough. No, because when you're dealing with these complex systems, here's a demonstration of sort of bifurcation of a far from equilibrium system. You may be able to get a probabilistic model, right? And even at the fundamental levels, the question is out. So whether quantum physics is truly probabilistic or not. But even if we accept that it's probabilistic at best, the sort of the divergence of probabilities is so fast and rapid that basically the amount of energy, time, information that's required to model a system completely, you know, exceeds any kind of, not even practical, but actual physical constraint. So that's something that we can, you know, has been shown by certain physicists. Specifically, I'm thinking of the work of Ilya Prigogine, the Belgian Nobel laureate who wrote a number of books. I think the English one, the one that's translated into English, or one of them is called The End of Certainty, where he kind of discusses how, you know, how practical, sort of the limits of practical modeling of thermodynamic systems, without even going into any quantum stuff, if I remember correctly. And we see this in biology, right? So organisms change their immediate environment, and that changes their evolutionary path. Termite mounds, the emergence of oxygen, the oxygen catastrophe early in the history of our planet, the development of biofilms and niche environments of any creature. And then there's the behavioral phenomena that are described in the ecological literature, like the Baldwin effect, where we talked about if an organism changes its behavior, and then that goes back and constrains some of the evolutionary pathways. And there's something called shielding, which is basically like the Baldwin effect. But Baldwin effect implies sort of a positive behavioral window opening up evolutionarily, or rather that evolution becomes sort of behavioral from a behavioral factor. And shielding is sort of the idea that the behavior then constrains certain development. So hygienic behavior may, for example, prevent the development of certain innate immunologic capacity, for example. And of course, humans demonstrate all these behavioral and phenomena in the way that we have evolved, right? Not that Baldwin, this Baldwin.
I was like, he looks too handsome to be a scientist.
So bottom line, we already talked about all this. Actually, we're not biological robots. Physical systems are not deterministic. In fact, they're probabilistic at best. And complex systems are non-reducible. And organisms experience, on some level, a subjective reality, and that's what allows them to evolve. You know, that's the basic idea. To quote Desjardins from The Phenomenon of Man, the engendering and subsequent development of the mind, in one word, noogenesis, when for the first time in a living creature, instinct perceived itself in its own mirror, the whole world took a pace forward. Man discovers that he is nothing else than evolution become conscious of itself, to borrow Julian Huxley's striking expression. So, yeah, where's the future hold? You know, it's just some ideas. We talked some about these scientific insights from dreams. Crick,
Yeah.
Kepler, Benzene, Mendeleev, the periodic table. He also ascribed to a vision. The idea of sleeping on sleeping on it to obtain irrational insights. And of course, we talked about Jung and Pauli's work. In the sort of the limits of computational modeling, right? What, again, going back to what we talked about in the introductory slides, the idea of combinatorial explosion, right? How do we rationally model a system which is ourselves? So again, you're dealing with a system has to be as complex as its environment to fully model. It wouldn't be a model then, it would be just a copy of the environment. So in order to model, we have to simplify things. And it may be that there is some other connection, some unconscious connection that allows us to simplify and evoke from nature true novelty, right? The unconscious, the transcendent function, the origin of novelty. And with that, I will end.
Yay! Ilya, this is fantastic. Quite the journey we've gone through. But this was fascinating, sir. I think I've already gotten out all of my brain's kind of spinning a little bit. I don't know if I really have anything intelligent to say at this point, but this is I need to process some of this. But this was very, very informative. Thank you so much for doing this.
Oh, it's my pleasure. I hope someone finds some value in it, or some insight, or some thread of
You have enough material here to write a book, I'd say.
well,
But so I'm kind of like, why haven't you done that yet?
there are a couple of books that have been written on a similar subject.
Oh, okay.
Shapiro, and I have one on my shelf by an Indian engineer in England who they don't quite combine the molecular genetics perspective. They're missing parts.
Right.
They're I tried to include as much broad category of evidence as I could, and also bring up enough specific examples to make it worthwhile. But there are many people who are thinking and writing about this, actually,
Yeah.
as it turned out. But for some reason, no one knows. I wonder why. As I mentioned, there's I think there's inherent fear and bias. The same reason people don't talk about Jung. People don't talk about Freud.
Yeah.
Unconscious, because that would challenge dominant view of our entire philosophy.
I think it would have serious consequences that we may not really fully realize just in the realm of the mind and how people structure their reality and understand things. It certainly smells like a Kuhnian revolution, if you're familiar with Kuhn.
Sure. Paradigm shift. Yeah.
Yeah. Yeah. And it's interesting how Kuhn points out, because I'm reading through that book, he specifically points out how the shifts that happen in science are always messy, and they always seem to have this strong element of, it's not really that any of the people from the previous paradigm are so much convinced as they just die out. It's a very morbid way to put it, but that he just points out that, you look at, say, Galileo's time, and it's like all these people disagreed with Galileo, disagreed with Newton, but the ideas were so fruitful that all of the younger generation really caught on to them, and it just sort of swept out in a generation.
And there's a natural defense to that, and that's the silo, right? The silo effect.
Yeah.
And that's the natural defense to this phenomenon. If you narrow things enough, you can sort of brainwash or enculturate the stagnation to the next generation. Yeah. And that's what happens. Yeah. I think.
It's interesting stuff. Before ending the recording, I wanted on the record that you've been tricked, because this whole time I've been psychologically typing you. I'm just kidding. That's just I do that anyway. But maybe we'll talk more after the recording. But I just want other people to know that I think INTP probably fits you pretty well. I don't know what you would think reading about that, but that's my intuition is INTP.
That is what I've scored on previous
Yeah.
test typing things.
Yeah, I think that makes sense. I'm sure a lot of people watching this, because of, you know, the tremendous number of examples, this won't mean anything to you, will say TE, but I've actually steered more away from that and think that's more NE, because it's actually very much like Darwin, who I do think was an INTP. But anyway, I just thought I'd put that on the record. But this was fantastic. Thank you, Ilya.
Absolutely.
Thank you very much. I will end the recording now.